So good afternoon, everybody. It is indeed a great pleasure to be here among such bright and creative minds to be able to discuss with you about what lies ahead in the future for science, and particularly for new technologies that we envisage would be important. So while thinking about it and route mapping for the future, the last item on my list, food, set the plant biologist in me thinking about what could be the challenges that plants or food crops in general would face in the future. And apart from the more canonical ones, or the predicted ones, such as the adverse environmental effects related to water scarcity, or changes in soil salinity, or temperature, or even the nutrient status of the soil, I feel that there would be a huge, severe pressure from biotic stresses in the future for the plants. And part of the strategy that we would need would be to finally balance the defense response in these plants as they come up with new pathogen challenges in the future. And to me, one of the most important traits for the future of plants, in the future for plants, or for future-ready plants, would be the ability to not just recognize, but to tackle these new pathogens in a very precise and highly controlled manner. In other words, this would mean enabling the plant to really finally balance its growth with immunity or defense in a very, very controlled manner. And I believe that understanding how plants communicate or integrate their external signals together with the pathogens will be very key in deciding how we go about designing strategies that would help us to improve the yield in crop plants in a very, very sustained manner in the future. And what my lab at TLL does is to understand the intricate chemical communication between plants to be able to decipher the mechanisms that would help us to combat them in the future. And what we use as a model pathosystem is the rice blast disease, which you see in the background, the complete devastation of leaves there, caused by the fungus called Magnaporta oryzae, which is one of the most devastating pathosystems known to crop production systems in the world. So what we do is to understand the intricate chemical communication between rice, one of its major hosts, and the blast fungus, Magnaporta oryzae, to understand how we could tweak the system into designing strategies for future plant protection. And we think that this chemical communication between the fungus and the plant starts fairly early when the disease papules, or these asexual spores called conidia, land on the surface of the leaf and start taking up cues that would allow them to finally make with what is one of the most incredible single cellular structures that I've seen. And this is the infection structure called the aprosorium that is made by this fungus on the surface of the leaves. And it allows the fungus to forcibly gain entry using very high hydrodynamic pressure to basically colonize the first few cells and then subsequently cause the damage that it is notoriously known for. I believe that the roots of the future lie in the past, and we use several classical molecular biology and recombinant DNA technologies together with some cutting-edge technologies to basically decipher the metabolomics status of both the fungus and fungal pathogen and the plant to be able to understand how they communicate with each other. And one of the powerful tools that we use is developmental genetics, using mutants that allow us to then have very clear insight into how these pathways work towards both causing pathogenesis on one hand and allowing the host to be resistant to the fungus. Recently, we have been involved in what has been an enigma for several decades now, which is to understand the trade-off and balance between growth and defense in the plants. So the idea in this case is to really understand at the molecular level how plants reallocate their resources during normal growth phase and when they're challenged by a pathogen or when they're about to start the resistance response, which invariably leads to a lot of cell death in the tissues that are now being colonized by these fungal pathogens. So this balance between growth and defense is something that we've been recently very interested in. And this is an unanswered question in plant immunity that has stayed on for decades now. And I'm really happy to announce that my lab has been very successful in gaining a breakthrough insight into this phenomena in a rather serendipitous manner through the study of a mutant which was carried out by an undergrad intern, Constance, in the lab. And the locus that she was working on is something called ABM. So what you can see here is the manifestation of a very novel form of defense in plants, wherein you have a very rapid response being built up in these black granules or inclusion bodies that you can see in the lower half of the image. And if you look carefully, you would see that these granules, or the resistance response, the novel resistance response, has the ability to really home in onto the fungus and basically decimate it within a few hours. So all you can see is the ghost of the fungus that has been left behind because of the manifestation of this novel form of defense. So what we had done initially was to use machine learning as an exercise to predict the occurrence of gene clusters in the fungal pathogen genome, which would allow it to perform secondary metabolism, because we believe that some of the chemical communication was happening through these secondary metabolites being produced in the fungus. And what was remarkable about this small gene that came up in this cluster, which is ABM, or antibiotic biosynthesis monooxygenase, it's an extremely short protein, just about 92 amino acids. Well, in fact, one of the shortest known monooxygenases at the moment. But what was remarkable about this gene was that it had come into the fungal lineage, in the blast fungus lineage, through lateral gene transfer or horizontal gene transfer from bacterial kingdom, and particularly the bacteria that colonize plant roots. So what you have is a remarkable occurrence of cross-kingdom signaling coming from bacterium into the fungal lineage that is now allowing the fungus to basically suppress the host defense through ABM function. Now, the next question that came up to us was, what exactly is ABM doing? I told you that it is a monooxygenase, but then my postdoc, Rajesh Patkar, who is extremely fond of rock climbing and is seen here about to reach the summit of one of the treacherous Agniban peaks in the Sahyadris, the Western India. And what Rajesh took up as a challenge was to identify what could be the substrate and the product of this ABM monooxygenase in the blast fungus. And through classical biochemistry and very sophisticated metabolomics, we were able to identify assays that would allow us to interchangeably swap these fluids from the mutant or the wild type and clearly see the suppression of defects in the mutant when you added the wild type extract to it, and vice versa. When you took the extract from the mutant and added it to the wild type, you would basically simulate the same novel defense response that I mentioned to you earlier on these black granules that you see. And through extremely intricate purification protocols using high-end fast performance liquid chromatography and tandem mass spectrometry, what we found ultimately were two fractions, one from the wild type and one from the mutant, that basically had the two activities that we were looking for. And believe me, the first half of these motivational messages that my collaborator bears of keeping calm is the more difficult part, given the sheer excitement that we had when we found these metabolites that are really the products of ABM monooxygenase. So what we found, in a short summary, was that ABM, which was now acquired from the bacterial kingdom into the fungal lineage, into the blast fungal lineage, was basically modifying the major defense hormone in the plants, which is jasmonic acid. And not just doing that, but it was making it into a metabolite that was inhibitory in nature. So it was hydroxylating it at 12 carbon and making it an inactive version. That not only caused the complete suppression of defense response in the rice plants infected by the wild-type fungus, but at the same time, it allowed the fungus to overcome the defense response and proliferate and cause the disease. The interesting part was that when you look at the mutant phenotype, when you lose ABM, the entire pathway shifts from making this 12-hydroxyjasmonate to making a methyl jasmonate derivative. Now, methyl jasmonate is the major trigger or the major metabolite that was the one that was causing the induction of that novel defense response that I mentioned to you about. So basically what we have are two extremely important metabolites now caused by a very short monooxygenase, one which is the inactive version, the 12-hydroxyjasmonate, and the other one is a methyl derivative, which is the more potent inducer of the defense response. What we have done subsequently is to take this basic research into an applied or a translational path, and we have developed some very intricate biosensors now that allow us to track the activity of ABM as well as the two metabolites that I just mentioned in real time in plants. So what you're looking at now are the FRET sort of analysis, where you can see the stability of a repressor complex that basically mirrors the presence or absence of ABM in these cell types. And what we do hope to do in the future is to see whether we could use this kind of monitoring of the defense response, not just in rice, but take it further to other crop plants as well. So what I've told you about is a novel defense response that is anchored on these inclusion bodies that we know virtually nothing about at the moment. And the main trigger for this kind of novel defense is methyl jasmonate or methyl jasmonic acid. And I feel that we would need all the bright minds, the really creative ideas, and great thinking, and out-of-the-box thinking as well, to take this further into the future to meet our goal of developing an extremely precise immunity response in plants. Thank you.